in my PhD thesis, I worked on the development of the immune system. And so my interest in developmental biology dates back quite a while. And I decided at the end of my PhD that I wanted to work on how the nervous system modulates immune function and immunity. So I had to do a postdoc in neuroscience and not in immunology. And then I moved to Tom Jesser's lab in New York at Columbia University and worked on the development of motor neurons, of the neurons that connect the brain with muscle. And from there I moved to the UK, so I haven't lived here before, obviously I have a German accent. And gradually, over the past few years, my lab has shifted from more basic questions in developmental biology of neurons to application of stem cell technology for the treatment of neuromuscular disease. So I basically moved over several relay stations to regenerative medicine, if you like. Yeah, so in the Jesser lab, I developed together with another guy called Hinek Wichterle a technology that allows you to directly derive motor neurons and also some other cell types from embryonic stem cells. And this technology we initially applied to developmental biology of motor neurons, but you can use very similar derivation methods to make, if you like, neuromuscular circuits in vitro and simulate neuromuscular disease. Yeah, so the best day in my scientific life so far was about 3 years ago when, in a collaboration with Linda Greensmith's group at UCL, we managed to graft stem cell-derived motor neurons into the peripheral nerve of a mouse, and then through light stimulation make these motor neurons control muscle. So this technology is very much in its infancy, but we think that long-term it may be used to allow patients suffering from ALS or also spinal cord injury to regain control of vital motor functions, such as the ability to breathe. So at the moment there are two themes in my lab. The first one is about modeling neuromuscular circuits and also neuromuscular disease in vitro with stem cell-derived neurons, glia, and muscle. So these are biochips, if you like. The second theme in the lab is to develop a new body-machine interface that will allow you to control muscle function through a biological interface and connect the host muscle, which in a paralyzed patient is no longer controlled by the patient's own nervous system, with an artificial optoelectronic pacemaker device. So it's about neuroprosthetics, really. Both types of project going on in my lab have a clear medical thrust to them, and we think that if we can get them to work, then the first set of project would be used for drug screens, in particular for drug screens for neuromuscular disease. And the second project would be used for the treatment of paralysis and for the reconstitution of motor function, which would be extremely valuable, say, to ALS patients who lose the ability to breathe as the disease progresses. Yeah, so at the moment I'm most excited about the fusion of biology, in particular stem cell biology and optogenetics, which is an artificial way to control biological function with light, with material science and optoelectronics. And obviously I'm quite a dilettante in the two other fields, so these types of projects involve collaborations with physicists and engineers. So things that I think will help to facilitate research and interdisciplinary research in particular is to find a common language, say, between biology and physics. My own experience with working with physics is that at least initially you need a translator because the same scientific terms have completely different meanings in the two fields. So if I had unlimited amounts of money, which I don't, then I would focus most of my efforts in moving this idea of a biological interface between neural implants and defective organs into reality. that would all involve, as we're doing now, the reconstitution of skeletal muscle function, but also the reconstitution of visceral motor circuitry, such as the circuits which control heart function and possibly also the function of the immune system. So most of the devices used today to stimulate nerve function and also muscle function are based on electrodes, and they're purely electronic. Electrodes have some disadvantages compared to a normal biological interface, because electrodes is just not the way normally different parts of your bodies communicate with each other. So if you could generate a more natural interface, namely if you want to control muscle, a synapse connecting nerves with a muscle, that would lead to much better and much more fine-grained communication between a medical device and the body, and would give you a lot more and a lot better control of a body function, such as breathing, for example.